While Parkinson's disease and Lewy body disease have different clinical presentations, the pathology is similar. These diseases are both synucleinopathies. They accumulate the same abnormal protein within cells that causes damage and kills the neurons. Lewy body disease is a progressive degenerative dementia of the elderly. Its primary feature is cognitive decline, particularly of executive functioning. It is the second most common form of dementia due to abnormal protein accumulation. Parkinson's disease is a movement disorder, but can also have disorders of speech, cognition, mood, behavior, and thought. Both these diseases are synucleinopathies. The synuclein family includes three known proteins: alpha, beta, and gamma-synuclein. Interest in the synuclein family began when alpha-synuclein was found to be mutated in several families with autosomal dominant Parkinson's disease. The function of alpha-synuclein is not well understood. We think it is located in mitochondria and has an important role in maintaining a supply of synaptic vesicles to the presynaptic terminals. It may also help regulate the release of dopamine. This neurotransmitter is critical for controlling the start and stop of voluntary and involuntary movements. Mutations in alpha-synuclein are associated with early onset familial Parkinson's disease. In both familial and sporadic forms, the protein aggregates abnormally. The same protein aggregates in Parkinson's disease and Lewy body disease to form Lewy bodies. Also present in Lewy bodies is a protein called ubiquitin. Its normal function is to tag proteins for degradation. As abnormally clumped alpha-synuclein does not degrade, it remains tagged. Parkinson's disease has been traditionally considered to be a non-genetic disorder. However, around 15% of individuals with Parkinson's disease have a first-degree relative who has the disease. We now know that at least 5% of people with Parkinson's disease have a mutation in one of several specific genes. The basal ganglia are the most seriously affected brain areas in Parkinson's disease. The main pathological characteristic of Parkinson's disease is cell death in the substantia nigra. Up to 70% of cells are affected by the time death occurs. Macroscopically, we can see depigmentation of the substantia nigra. The underlying pathological defect is damage and death of the dopaminergic pigmented neurons of the substantia nigra, which are the cells of origin of the nigrostriatal pathway. Illustrated here in post-mortem specimens, the gross change is a loss in pigmentation of the substantia nigra. Microscopically, we see neuronal loss and gliosis. In addition, there are cytoplasmic inclusions, the Lewy bodies, seen here as the pink spherical objects within the neurons, and in the inset picture as the body stained with an immunohistochemical marker. In this higher powered view of two neurons, we have a normal neuron on the left with pigment in it and no inclusions. it has long dendrites. The lower right contains a neuron in which there is a prominent Lewy body. It is retaining some pigment at the moment, though this will be lost in future. Lewy body disease. The cause of Lewy body dementia is unknown in most cases. Inherited cases comprise about 10% of all those diagnosed. The primary feature is cognitive decline, particularly of executive functioning. Executive functioning allows us to plan, analyze, and engage in abstract thinking. This is lost in patients with Lewy body disease. One of the most distinctive clinical features is hypersensitivity to neuroleptic and antiemetic medications. Lewy body disease is characterized by Lewy bodies throughout the brain. There is a loss of dopamine-producing neurons in the substantia nigra, similar to that seen in Parkinson's disease, and loss of acetylcholine-producing neurons, similar to that seen in Alzheimer's disease. In Lewy body disease, as well as acquiring the Lewy body inclusions, the affected neurons also acquire neurofibrillary tangles similar to those from Alzheimer's disease.